Sure. Do you see my screen?
Yes,
Okay,
it's coming in and I can hear you just fine.
great, perfect. Yeah, so my name is Mike MacCoss, and I run a proteomics lab at the University of Washington. And one of the things that I'll point out, we've been collaborating with LabKey. I think it's now like 9 or 10 years, and they're members of my lab that have been working with LabKey since its origin. So that's partly how the connection got started. I kind of want to bring up a little bit about how our mass spectrometry lab uses Panorama and LabKey, and I'll explain a little bit about what Panorama is to manage our mass spectrometry raw data, our process results, and our collaborations. And I'll point out that this is definitely a work in progress and something that's constantly being improved. It says that's one of the great things about working with LabKey is that things are constantly improving. It's a active project under development. And that's one of the things we find particularly satisfying with working with LabKey. Okay, so just before I start with Panorama and LabKey, I need to talk a little bit about the client tool Skyline that our lab developed and why we reached out to LabKey to interact with them. So we built this tool, Skyline. It's being managed by Brendan McLean, and we have a fantastic development team working on this. And it's a Windows client. It started off being very protein peptide centric, but we've kind of extended to being analyte agnostic. It's very intuitive, document editor, graphical user interface. It's got a lot of undo redo. It reads all the mass spectrometry vendor data natively without conversion. It's free and open source. It's developed and supported professionally. And the key thing as well is that we actually do work very closely with these instrument vendors and make sure that we support their platforms as best as possible. And I think that's kind of key. I think you'll see that quite regularly. We do like to interface with the instrument vendors, but now we're trying to think about how do we actually store and manage these documents. So when this started, we would go to places and don't get me wrong, we love this client tool Skyline, and it's really kind of the key for interfacing with the mass spectrometer. But we wanted this ability to store and manage these documents. We wanted them to ultimately be queryable and findable. We wanted them to be in a relational database where you can control the security of the data. And so we built this tool. It's Panorama with LabKey, and it's a module within LabKey Server. It's designed for sharing Skyline documents and the associated data. We actually offer a free and hosted version at the University of Washington. It's called panoramaweb.org. And right now there's about 565 labs hosting their data that are hosted on this server. We have, a fairly large number of Skyline documents there. The users themselves control the security of it. It has all the complete features of a LabKey server, and we find it very, and I'll give you examples, excellent use of being able to share data between labs in kind of consortium wide projects, and also at the time of publication and ultimately making data fully public. We also have this portion of this Panorama web server, which we call Panorama Public, and I'll talk a little bit about this towards the end. But ultimately, one of the problems with any sort of omics type data is you have to make it findable. So the way the proteomics community does this is they have these IDs called the proteome exchange IDs. And these are indexed between several different repositories. So when you publish a paper, you also have a proteome exchange ID. One of these servers that hosts this data is our Panorama web server, and there are currently about 320 public data sets there. And you can kind of keep it private during the review process and ultimately make it immutable and when it becomes public. One final thing I'll point out is while we host a version of the server, it is also locally installable. And we found that there are a number of companies in particular that like to have host their own version of it. And I will encourage you, if that is a solution that you would like, there is this a program we have called the Panorama Partners, and it really is a partnership that's kind of joint between LabKey, our lab, and also the partners who are the people who are kind of who want a support contract, but at the same time are providing kind of the main driving influence and use cases for a lot of the new features that get added. Okay, so in Skyline, I'm showing kind of a very simplistic form of this right now, but you basically have your list of peptides and information and your underlying processing data. You can use this to kind of quantify your peptides or your analytes, and you can actually publish this and just directly from the client tool push this up to Panorama or as a way and store and manage it. You can browse which folders you have access to. And again, a lot of this is all, the security is all controlled entirely by the user on the server side. So the way I like to think about this is we have this client tool. The data can get pushed and published to Panorama. You can then use it in the context of this, this relational database. You can use R and Excel and generate different data reports from that. And of course, then you can then share these documents with other people where people can download them and access them using the client. There is actually a nice web UI that people can access a lot of this information as well, and I'll show some of that, et cetera. So if you're an academic lab, you can get access to this server for free. You just request a lab account. Currently there's about 565 labs. It's kind of growing in this linear fashion. And the Skyline documents are also growing at different rate. And we have different types of documents, like those that are for different experiments and quantitation, those that kind of build libraries. And one of the things we found is that instrument system suitability and quality control, it's something that's partially driving this usage. Allows you, and I'll talk in a second about this, to track this information longitudinally over time. So just to kind of give you a little bit of some of the views, one of the things that's kind of nice, LabKey Server is incredibly flexible, and one of the things that has been a joint effort by LabKey and our lab is to try to make it so that there are some very specific quantitative proteomics views. And so you can kind of look at things at a document level and see these are all the proteins and the underlying peptides. And just like with a regular LabKey server, most of these fields are adjustable and change. You can alter the views, alter which columns and views are present. You can sort them by any number of different ways. You can generate charts, you can export it, etc. You can also view underlying information within proteins, and then with those proteins, you can underline the view of peptides. You can actually get things like quantitative peak areas, and you can, there are lots of just generic default plots. A lot of these mimic some of the plots that we currently have in the client tool. You can also view a lot of the underlying peptide data directly all within the web interface. You can look at different things like the ratio of the unlabeled to the stabilized to labeled internal standard. You can generate a ton of different plots. You can group them by condition, etc. So these are just a lot of the capabilities that are within the web browser, and it's something that we're very kind of excited about seeing this continue to grow and expand. And we've noticed that people have started to do bigger and bigger experiments. We've got some ideas in the upcoming years on how to make that a little bit more manageable within the Panorama UI. So one of the things that's happened in the last couple of years is that we spent a lot of effort in making sure that Skyline, the client tool itself, had tracked all the changes in an audit log. And so this that every action, every altering of a chromatographic peak is all tracked in a fairly detailed way. It's fully internationalized, so depending on what language your computer settings are set at, you will display the audit log in that native language. And if you actually upload the Skyline document to Panorama, that information is all entirely tracked as well. So you can get access to the full audit log of the document without having to download the document. And while we're not quite there yet, we're definitely have this, we're getting close to what we believe is an FDA 21 CFR Part 11 compliance. This is definitely a ongoing request that we frequently get asked by for some of our partners, and this is something that we're pretty excited about. And you can, again, with all of these lists that you normally have in LabKey Server, you can sort them, adjust them, you can change the views, but all this data is managed within the relational database. We often find that people like to have their calibration data, including our lab. We spend a lot of times often building a quantitative assay, and the validation of that assay and the figures of merit that we want to have are things that we like to be able to store and manage within the relational database that people can go back to over time. And here's just an example of some of the views that have been added specifically for this. So one of the things that our lab got really excited about was this idea of adding like another wrapper around our Skyline client, which we call Auto QC, which would actually sit on the instrument computer. And as a folder sees new things coming off, it would automatically check the file folder. It would import the results into a Skyline document, and then it would process those results in the Skyline client and then pass that up to the Panorama server. So this became an automated approach. It allowed fairly extensive ability to track data longitudinally that's being collected in real time on the mass spectrometer and imported into Panorama. And it's also vendor agnostic. So it doesn't matter what platform you're on, it's looking just for changes in the files that are being posted to a specific directory on the local client. So we use this for a couple of different things within our lab. One, we use it for system suitability. So how is the system, how good is the mass spectrometer working? Can we track this over time? And also we have this thing we use as process controls, which is for a specific sample, did it get prepared and is it working okay? And did the sample preparation work okay? Did we have a bad batch of trypsin? Did the sample prep, did our solid phase extraction just not work correctly, et cetera? And in that case, we add an intact protein prior to the sample preparation. We add a set of peptides at the last step. So that one's used to assess the system and one's used to assess the sample prep. And we use this auto QC strategy for both of them. So the first step with system suitability, we pick basically a sample that's stable, don't change over time, minimal carryover, inexpensive. And in our case, we wanted to be able to assess that the peptide intensities are within tolerated variance. We want to be able to use to assess our chromatography, and we want to track mass accuracy. And I'll show you examples of this over time. We tend to use a mix of a commercially available retention time calibrator from peers. We also use a digested protein just to give us some extra peptides. It's really inexpensive and reproducible for us to produce. And also we use this to assess lots of different things. This is a paper that Michael Behrman had a number of years back and was kind of some of the inspiration for this auto QC folders within Panorama. But we basically take a lot of inputs from within the mass spectrometer, and we basically have a bunch of metrics, and we can use this to make Levy-Jennings plots, Pareto analysis, et cetera, across time. And this is basically how it works. Every X number of samples, we run this system suitability sample. This is what the client tool that sits on the instrument looks like, and basically provides some sort of authentication to Panorama. And then as it sees a new file being written, it basically opens that file, imports it, and posts it to the correct Panorama folder. You can get lots of different views of the data, and I'll show you some different examples of this in a bit. You can display things like changes in peak area, mass measurement accuracy, chromatographic performance, et cetera. And you can also track these things over time. And I can imagine these same sorts of things would be very useful in the context of like biologics. In fact, we have some of our colleagues who are thinking about using these things for biologics, right? We use it for tracking the performance of a system, but you could also think about tracking it between batches and sample preparation of a biologic material that's prepped in different lots. We can kind of get this overview of the system. You can kind of see what the status is from this dashboard, and we can track things like we call it precursor product ion ratios or precursor transition ratios, and this tends to gives us a good indication whether or not we have things like a dirty quadrupole, et cetera. We can also start to see tracking things like mass accuracy changes over time. And that may tell us that we need to actually recalibrate the instrument. And the way we treat this is that this is something that is a lab-wide folder for each instrument, and it is the responsibility of each user of the instrument, whether it's graduate student or postdoc or staff scientist, to basically use this and track the system suitability while they're using it, so that when a new person comes up to use this instrument, they have some sort of record of what's happening. And I'll point out that this is extremely valuable with working with the instrument vendors. It allows us to show how an instrument may be changing over time. It allows us to be able to have a quantitative set of metrics of when an instrument's not working ideally. Okay, so this process control. So in this case, we use a yeast protein and allows us to kind of assess different steps of the preparation. If something happened to the digestion step, we'd see problems with the enolase peptides, but not these PRTC peptides, these retention time calibrator peptides. If something happened to either the steps after the digestion, you'd see issues with both the enolase and the PRTC peptides. So when we store a set of each member of the lab who's producing, participating in data production efforts, we end up having a project folder for that project. And within each project folder, there is an experiment subfolder. And the way we currently do this is just using a lot of LabKey server type methods, where we have wiki pages that provide some sort of description. And within this, we also have a set of subfolders. And within this subfolders, one of them is normally a process control folder, where we display and show the results of specific peptides that were spiked into each individual sample over time. And you can see from some of this data that there may be an issue with this one sample that occurred on April 29th, and that there's clearly a deviation in some of the peptides that they're relative to the other ones. And that allows someone to get this 30,000 feet overview on whether or not something may be wrong with an individual sample, and whether or not you want to omit it, or whether or not you want to go in and inspect it a little bit more detail. We also then, for that same experiment, have all the individual quantitative data. And in here, we host the Skyline document, and we also host all the associated raw files. You can actually click into the Skyline document, and you can either download it and look at it in the client, or you can look at it within the web browser. I also kind of want to point out briefly is that a lot of the metadata currently is kind of just stored as kind of flat information. You can imagine we're very excited about the LabKey sample manager, which I think you're going to hear a lot more about tomorrow. We really are kind of desperate to have all of this linked, and I'll show an example of this at the very end, where we have the data where we can take it from where it's actually not stored as kind of just flat information in an unorganized manner, that's actually stored in more of like a LIMS type sample tracking information. We do also use this a lot for different collaborative projects. So this is a collaborative project funded by the NIH, and the NIH loved the fact that this project basically could be branded on itself, all within the same Panorama web server. We can generate short URLs for this. We can have multiple different projects. This is between our lab and Annie Hoofnagel's lab at the University of Washington, Jenny Van Eyck's lab at Sinai, and Wei Jun Hyun's lab at Pacific Northwest National Laboratories. And we can kind of provide just the normal information like, news feeds, we can have message boards, et cetera, all within the project. We can have a public facing portion of it and a closed security portion of it. We can, under different projects, we can have different experiments. We can dig into it. We can, obviously have different wiki pages and share information as public. We can actually have the underlying mass spectrometry data and some visualization of the quality of the data and the results are being presented. And we've done this for a number of different projects, and we definitely encourage people to kind of reach out to us if you have a project like this and you'd like to host it on Panorama. This got brought up by Adam Rausch yesterday, but one of the that's kind of this strength of using LabKey Server for hosting and managing your data is just the API interfaces. And we've worked with a number of different projects. One is this National Cancer Institute CPTAC assay portal, and another one is this NIH Common Fund Links project, where the data is actually physically sitting in our LabKey server, but the data has a different view and an image that pulls the data from our server in real time to analyze it. I'm kind of beginning to get towards the end, but the other thing that we like the fact that if the data is hosted in this Panorama web server, it's fairly easy to make it public and immutable at the time of publication. And then ultimately within the academic community, at least, that data becomes findable. We generate short links, we can generate proteome exchange IDs, we can even generate DOIs. And so that the data, regardless of what proteome exchange server is found on, is present on, you can actually get access to it within our server. It also becomes read-only and immutable. So that's something that's a kind of a critical component for the journals for publication, that the authors and the data generators don't have access to the data and can't change it or remove it over time. And we currently have, this is what that page looks like. You can go there and explore the different Skyline documents, the raw data, et cetera. And this is the last part. I'm really excited about. This is something we've been mentioning to LabKey for a number of years, and it's finally coming to a fruition. And that is, we really want to be able to link the results with the actual samples. And with the LabKey Sample Manager, this is definitely now going to become possible. And so we want, for samples in the Sample Manager, we want to be able to go to a sample and to be able to see any of the Skyline documents in Panorama that contain the results, and kind of showing this on the right-hand side. We also are not there yet, but we'd love to be able to see the opposite, right? Which is, for analyses in Panorama, you'd like to be able to see results linked to the metadata, where the sample locations may be, what freezer spots they may be, where can I find them, is there any of that sample left, et cetera. And then we also want to be able to make it easier to get the metadata and the groups and the labels that can be stored and managed in the Sample Manager into our Skyline document, that client tool. And that's definitely something that is in the plan for development, but it's something that is particularly exciting when all of these two components can kind of come together and kind of and complete the cycle. Okay, so a little bit about this ultimate vision, and that is, we'd love to be able to host all the information about our samples, metadata, preparation, location, et cetera, in one server. We're completely all in on using LabKey Server. We have this Panorama module for storing and managing targeted proteomics data, and it's kind of been extended to just all mass spectrometry data that's been analyzed with Skyline. And use that information to ultimately design experiments, to track the system suitability and process controls, like I mentioned with you, to add metadata to Skyline that's stored potentially within the Sample Manager. So that's not correct. It actually should be the Sample Manager, not the Panorama module. We should want to be able to think about, potentially, are there steps that we can batch process, computationally expensive steps? And that's again something that we've had discussions with LabKey about. We'd like to be able to manage this process results. I told you a little bit about how we track the audit logs, but it also version controls the Skyline document. So if you're continually posting the same document back to the LabKey server, it will keep all the older versions of that document, and you can revert to the past ones at any time. You can control the data access, and data is ultimately findable upon publication. So with that, I do have some acknowledgements. So in red on the left-hand side is the University of Washington, my lab at the University of Washington. And in red is actually the Skyline and Panorama development teams on our side. But this is entirely a huge collaboration with LabKey. This wouldn't be possible without that connection. Brendan McLean, in particular, who was a co-founder of LabKey, initiated this discussion. I remember when he presented it to me, thinking that this was right up LabKey's alley, and it's been a fantastic interaction. Vigisha Sharma has been kind of the development lead for Panorama on our side, but any of these people in red have definitely worked on this this interaction and project over the years. We're thankful, of course, for the federal government for helping us provide some financial support. The instrument vendors do provide support for the client and that interface the client to LabKey. And I want to give a quick shout out to the Panorama partners. This is truly is a partnership. And Michael mentioned yesterday during his open talk that LabKey is different. And I think this is kind of an example of this. This is kind of a model where people come together, not necessarily to compete with one another, but instead they actually come with a common set of needs and they build off of one another. And a lot of times people gain access to development features that another one of the partners had suggested and prioritized. And I think this has been kind of an excellent case where different either pharmaceutical companies or instrument companies or even the National Cancer Institute have benefited largely by the development efforts of another one of the partners or development priorities of another one of the partners. And I'll thank you very much for your attention, and hopefully if there's a little bit of time, I can take some questions.
Yes, thank you so much, Mike. That was very enlightening. Anybody have any questions for Mike MacCoss? Give people a minute to digest all that.
I recognize that it may be a slightly different use case, but I think that is a growing area, and I can see parallels definitely with the aspects in the biologics. I think there's a question here.
Let's see.
Yeah. So
yes. Go ahead.
I think there's a question I see right here. So this is, I can repeat here. So where should mass spec labs get started with Panorama? Those using Skyline and those not using it. So I think the key thing is to start with the panoramaweb.org server. You can request an account. There are some tutorials. There's actually some great saved webinars up there and videos. So panoramaweb.org. It's hosted at the University of Washington. It's completely free to use. And there's a lot, if you have questions and feedback, there's a support board that's monitored, et cetera, from that. If you're not using Skyline, the first thing we would try to encourage people to do is to try it. There's a skyline.ms is the website. It's kind of easy to remember. It's become one of the most widely tools for the quantitative analysis of mass spectrometry data. I think we're getting on the order of about 1,000 installations per month or per week, and there's about 10,000 boot ups per week as well. So this is definitely a growing area, and there's tens of thousands of users. So if you haven't used it yet, definitely familiarize yourself. There is tons of documentation for it on the website.
Let's see, we do have a question here. It's maybe not so much a question for you, Mike. It's about parallel capabilities in the space of genomics gene expression data. I'd invite people to engage with us about what they need in that regard. We do have some people that use LabKey for that purpose. So let's talk about that a little bit more. Let's see.
I can begin to answer the last question a little bit. We have gotten a fair amount of requests about using things like AutoQC to track longitudinally differences using mass spectrometry of the biologics that are being produced. So basically using the mass spectrometry as an assay type. There's definitely been a fair amount of development by LabKey on the multiple attribute model. And this is, and also in DMPK, so drug metabolism and pharmacokinetics analyses. They're actually, I didn't show those because our lab are not widely users of those, but those are some incredible views and displays that are currently present within Panorama. And I know that there has been some discussion within the discussions about the biologics and linking that. but I will let Bernie maybe talk a little bit more about that.
Sure. So Mike showed what the current work will kind of look like between Sample Manager and Panorama, and actually it's going to be the same between Biologics and Panorama. It's going to be an iterative process where we want to learn what is most useful to the organizations that are taking advantage of this. But that is step one in integration between Panorama and both Sample Manager and Biologics. And once we have that available for exploration, we'd love some additional feedback from other organizations. Thanks for asking.